Hello, everyone. My name is Arun Jani, and I'm a postdoctoral associate in the Horticultural Sciences Department at the University of Florida. And today I'd like to share some of the research that I've been involved in exploring how sweet orange planting configurations and nutrient management strategies can affect the productivity of young groves planted in the Indian River District of Florida. Now, the Indian River District, for those of you folks who don't know, is located on the east coast of Florida, and it is a region that has historically been dominated by grapefruit production. In fact, 79% of total grapefruit produced in Florida is grown in the district, while only approximately 3% of total orange production is grown in the region. However, growers are looking to diversify and expand into orange production. And that's really with good reason, because previous rootstock trials in the district have shown that sweet orange fruit yields and quality can be very competitive in the region. It's also important to point out that there is a trend all over Florida, and the Indian River District is not excluded from this trend, to plant at higher density groves to optimize earlier yields before trees succumb to the bacterial disease of Huanglongbing. For those of you folks not familiar with Huanglongbing, this is a devastating bacterial disease that's present throughout the state, and the bacteria clog the phloem of trees, preventing carbon translocation throughout the plant, which really leads to a reduction in root growth in the trees, and trees that have a severe disease incidence can become unproductive or die within a matter of years. So it's really important to think about tree density as we think about nutrient management strategies for citrus. And one of those nutrient management strategies that can be used and can be coupled with higher tree density is the advanced citrus production systems. Now, ACPS is an integrated system of irrigation, nutrition, and horticultural practices that provide control over tree growth by using irrigation to train tree root systems to a limited area and to fertigate frequently. Now, in Huanglongbing affected trees that have reduced root systems, the ACPS system can be highly effective because irrigation and nutrient resources are directed toward the root system. Now, ACPS concepts have been applied to orange groves in central and southern Florida already. And what we found is that up to 100% higher inorganic nitrogen or plant available nitrogen in the irrigated zone with daily or weekly drip irrigation is possible relative to monthly irrigation using wide pattern micro sprinklers. Our colleague Morgan et al. Also found that frequent fertigation, up to 30 times annually, can result in larger fruit trees and yields than infrequent fertigation or the use of controlled release fertilizers. However, we need more information regarding how ACPS can affect the orange grove production in the Indian River District in conjunction with higher planting densities. So that brings us to the goals and objectives of this research. Our primary goal here was to identify a set of management strategies that Indian River District growers could feasibly use to optimize the productivity of young orange groves in the region. Our specific objectives were to evaluate how different combinations of production practices that included tree density, fertilization management, and irrigation would affect, number one, tree growth and foliar nutrient content in young Valencia sweet orange grove. And then number two, how these factors would affect fruit yield and quality during early years of production under HLB endemic conditions. So our study was conducted at the UF-IFAS Indian River Research and Education Center in Fort Pierce, Florida. We conducted this research on a soil typical of Florida coastal soils where citrus is produced. The soil was poorly drained and it had low inherent soil fertility. And of course, we conducted this research under typical subtropical environmental conditions of Florida. The experimental setup consisted of Valencia sweet orange trees planted on Guarski citron rootstock in September 2013. We utilized a randomized complete block design with 8 replications, and we implemented 3 unique treatments. Treatment number 1 consisted of standard tree spacing which was at 357 trees per hectare with controlled release fertilizer, or CRF, and micro sprinkler irrigation. Now, we used 1 emitter per tree to irrigate, and we applied fertilizer at the rate of 224 kilograms of nitrogen per hectare. Our second treatment consisted of a high-density planting of 953 trees per hectare, fertigation, and micro sprinkler irrigation. Now, here we used 1 emitter to irrigate 2 trees, and we fertigated weekly at 224 kilograms of nitrogen per hectare. Treatment number 3 also consisted of a high-density planting with fertigation, but instead of a micro sprinkler system, here we used drip irrigation with 2 drip lines applied per row. And then again, fertigation was also applied weekly at 224 kilograms of nitrogen per hectare. So these 3 treatments were compared in this study. Now, this is an example from the field of treatment number 1, the standard tree spacing with controlled release fertilizer and micro sprinkler irrigation. We had 2 rows per bed. And then this is an example of treatment number 3, which again consisted of high density planting, fertigation, and double drip lines, where we had a staggered planting, the equivalent of 4 rows per bed. Let's take a look at some of the results, first focusing on tree growth parameters. Now, on this figure, we have trunk diameter on the y-axis. We have data presented over 6 years, beginning in 2014 and ending in 2019. We have our 3 different treatments, which are color-coded. The bars on the graph, the color of the bars correspond to the treatments on the x-axis. So we have treatment number 1 in blue, treatment number 2 in red, and treatment number 3 in green. And what we can find here, if we focus on from 2016 onward, we see a consistent trend of higher trunk diameter or larger trunk diameter with the standard tree spacing, controlled release fertilizer, and micro sprinkler irrigation compared to our 2 high density planting treatments. And this is most likely due to the greater space available for lower density plantings and the lack of competition relative to higher density plantings. And on this next figure, we look at canopy volume on the y-axis measured in cubic meters. And again, we see the same trend that we did with trunk diameter. We can see if we start at 2016 and move onward, we can see a general trend of higher canopy volume with treatment number 1, which again consisted of that lower density spacing relative to treatments number 2 and 3, which consisted of higher density plantings. And again, these data can likely be explained by the lower competition for resources, water and nutrient resources, with lower planting density. Now what I'd like to do is move on to foliar nutrient concentrations. Now, we collected foliar nutrient concentrations over 6 years, beginning in 2014 and ending in 2019. And here we're focusing on macro and secondary nutrients: nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. And on each of these small figures, what we can see is a horizontal slashed line, and that corresponds to the optimal nutrient range. And really the key point to point out here is that in most cases, nutrient concentrations were either in the optimal range or were higher than the optimal range. And this indicates that all 3 treatments used in this study were adequate in meeting tree nutrient demand. And it's important just to point out at the bottom right-hand corner of the screen, optimal ranges for sulfur are not available, which is why they are not placed there. Now let's move on to our fruit yield parameter results. What we'll start with is reporting fruit yield on a tree basis in kilograms, and we have data presented for 4 years, and our final figure on the far right is cumulative yield data. And what I'd like to do is focus more on the latter years, so 2018, 19, and 19, 20, as the trees grew a bit larger. And what we can see here really is really a lack of consistency. we can see that in 2018-19, the standard tree spacing performed the best on a per tree basis. But then in 2019-20, we saw no differences between any treatments. And it's also important to point out, folks, that in 2017-18, starting earlier on, we had low yields due to Hurricane Irma damage. But now moving to the far right and looking at cumulative yield data, what's important to point out here is that there were no statistically significant differences between treatments on cumulative yield data, indicating that all of these treatments led to similar fruit yields per tree when considered over a cumulative 4-year period. Now let's look at fruit yield on an area basis. Now what we can find here is that higher tree density resulted in higher yields on an area basis, which is not all that surprising due to the drastically higher tree density of 953 trees per hectare compared to 357 trees per hectare at the standard tree density. And again, let's focus on cumulative yield data here, folks. If we look at cumulative yield data, what we can see is that over a 4-year period, we had substantially higher cumulative yields on an area basis with treatments 2 and 3, high density plantings with fertigation, with either micro sprinkler irrigation or double drip line irrigation compared to treatment number 1, which again consisted of that standard tree spacing, controlled release fertilizer, and micro sprinkler irrigation. Now let's move into the yield of solids. And again, if we look on a tree basis, we can see a general lack of consistency when it comes to results. In 2018-19, as the trees grew a bit larger, we can see that yields per tree were actually higher at the standard tree spacing treatment relative to the 2 high density treatments. However, if we look cumulatively, we see no significant differences per treatment cumulatively over the 4-year period. And again, as a reminder, folks, in 2017-18, yields were low due to Hurricane Irma damage. And if we look at our yields of solid data on an area basis, we see the same trend that we did with fruit yields. We see that higher tree density usually resulted in higher yields of solids. So if we look at our cumulative data on the far right, we can see that treatments 2 and 3 led to yields of solids, which were substantially higher than our standard tree spacing of 357 trees per hectare with controlled release fertilizer and micro sprinkler irrigation. And what can we conclude from this research? Well, let's start with our tree growth data. What we found was that trees planted under standard tree spacing, controlled release fertilizer, and micro sprinkler irrigation grew larger than trees planted under the 2 higher density and intensive fertigation and irrigation treatments. We saw that fruit and solid yields per tree, in most cases, were not higher under standard tree spacing compared to high density treatments number 2 and 3, even though the high density trees received less nitrogen fertilizer per tree. And that's an important factor to point out, folks. Remember, we applied 224 kilograms of nitrogen per hectare with all treatments. And that means that trees planted at higher density received less nitrogen per tree than trees planted at the lower density. Yet in most cases, fruit yields and solid yields were not affected. Fruit and solid yields on an area basis were generally higher under the high density treatments relative to the standard tree density treatments. And then finally, if we look at our nitrogen rates used in this study for standard and high tree density groves under HLB endemic conditions, we can find that they need to be reevaluated. As I previously mentioned, the yields per tree generally did not differ, even though all trees receive the same nitrogen fertilization rate. So we need to reevaluate nitrogen rates for high density groves. And finally, folks, I'd like to thank the agencies that funded this research, USDA NIFA and the UFI for Citrus Initiative, as well as our donations, the companies that provided our fertilizer for the study. and all members of the Ferrarezia Research Lab. And with that, folks, I would be happy to answer any questions during our Q&A session. Thank you very much.